Welcome to Pharma World Channel. Today we're taking you on an exclusive tour about the GMP requirements of the facilities, premises, and critical utilities that make every single dose safe and effective. We will go deep of the main facilities of the pharmaceutical factory, warehouses, production and quality control, along with the required utilities for pharmaceutical manufacturing operations. A pharmaceutical facility isn't just a building. The premises, utilities, and equipment must be designed, located, installed, and qualified to facilitate cleaning, maintenance, and operations, according to the dosage forms to be manufactured in and the stage to be carried out. The layout and design must aim to minimize the risk of potential contamination and, in general, any adverse effect on the quality of products. Let's break it down. The pharmaceutical factory is divided into separate, controlled premises and facilities to prevent cross-contamination as follows. Warehouses. The first main area in the pharmaceutical factory is the storage areas, which are called stores or warehouses. It consists of two main storage areas. The first one is the materials store, in which the raw and packaging materials, and it includes the following 7 sub-areas. 1. Receiving area. This area should be designed and equipped to allow containers of incoming materials to be cleaned where necessary before storage. 2. Quarantine area, where quarantine status is ensured by storage in separate areas. These areas must be clearly marked and their access restricted to authorized personnel. Any system replacing the physical quarantine should give equivalent security. 3. Sampling areas. Normally be a separate sampling area for starting materials. If sampling is performed in the storage area, it should be conducted in such a way as to prevent contamination or cross-contamination. 4. Released materials areas. Segregated areas should be provided for the storage of approved materials for production, according to the required storage conditions. Printed packaging materials are considered critical to the conformity of the pharmaceutical products, and special attention should be paid to the safe and secure storage of these materials. 5. Dedicated areas for special storage conditions materials such as cold chain, 2 to 8 degrees Celsius, refrigerated, -20 degrees Celsius, freezers, -70 degrees Celsius, ultra low, special humidity for hard gelatin capsules, if required by the manufacturer of the capsules. 6. Restricted access area of the high-risk materials, narcotics, controlled substances. 7. Rejected materials areas, a separate and limited access area for rejected materials, prior return to the supplier or destruction. The second main storage area is finished product store, where the products received from the production and include the following 8 sub-stores. 1. Receiving area. In this area, the store team will receive the products after completion of all manufacturing processes. This area should be designed and equipped to avoid contamination and cross-contamination of the production areas. 2. Quarantine area. When the product received from production, it is not ready for distribution in the market, where quarantine status is ensured by storage in separate areas. Prior confirmation the compliance of quality and the safety of the batch. This area must be clearly marked and their access restricted to authorized personnel. Any system replacing the physical quarantine should give equivalent security. 3. Segregated areas should be provided for the storage of approved batches for distribution. 4. Individual dedicated areas for free samples, returned and near-expiry products if available. 5. Recalled product store. In the event that a quality defect of certain batch or batches, limited access store is required for their temporary storage until the decision as to their future use has been taken, needs to be investigated or a batch recalled. 6. Dedicated areas for special storage conditions products such as cold chain, 2 to 8 degrees Celsius, refrigerated, -20 degrees Celsius, freezers, -70 degrees Celsius, ultra low. 7. Restricted access area of the high-risk products, narcotics, controlled products. 8. Rejected products area, a separate and limited access area for storage of rejected products prior decision of destruction or any other decision. Production. The second main area in the pharmaceutical factory is the production areas, which is the most controlled areas in the pharmaceutical factory. Access to production premises should be restricted to authorized personnel. The production areas are generally consisting of 3 main units. The first unit of production is changing areas. It should be designed as airlocks and used to provide physical separation of the different stages of changing in order to minimize microbial and particulate contamination of protective clothing. The changing areas should be flushed effectively with filtered air. The final stage of the changing room should be the same grade as the area into which it leads. The second unit of production is the manufacturing and primary packaging areas. The core concept is zoning based on product and process criticality. Weighing of starting materials usually should be carried out in a separate weighing room designed for such use. For oral solid dosage forms like tablets, are classified environments, like ISO 8, grade D. The risk is contamination from other products or microbes. These areas are well controlled but don't require the highest level of sterility. You'll see large open bays for granulation, compression, and coating, all designed for easy cleaning. Now, for injectables and eye drops, the stakes are infinitely higher. Any contamination directly entering the bloodstream and mucous membranes can be catastrophic. This is the domain of cleanrooms. These are classified environments like ISO 5, grade A, where the number of airborne particles is controlled to an incredible degree. Access is severely restricted, and operators wear full sterile gowns. For semi-solid products like creams and oral liquids like syrups, the focus is on microbial and chemical contamination control. These areas are hygienic and often have dedicated equipment to prevent cross-mixing of powerful odors or ingredients. The manufacturing and filling areas are classified environments, like ISO 8, grade D, unless required higher classification are sterile creams where it will match injectable products. Technical measures. The following 9 techniques can be addressed based on the nature of the products. 1. Dedicating the whole manufacturing facility or a self-contained production area on a campaign basis, dedicated by separation in time, followed by a cleaning process of validated effectiveness. 2. Keeping specific protective clothing inside areas where products with high risk of cross-contamination are processed. 3. Use of closed systems for processing and material product transfer between equipment. 4. Use of physical barrier systems, including isolators, as containment measures. 5. Dedication of equipment, dedication of product contact parts, or dedication of selected parts which are harder to clean, e.g. filters, dedication of maintenance tools. 6. Use of single-use disposable technologies. 7. Use of equipment designed for ease of cleaning. 8. Appropriate use of airlocks and pressure cascade to confine potential airborne contaminant within a specified area. 9. Use of automatic clean and/or sterilization in-place systems of validated effectiveness. The third unit of production is secondary and tertiary packaging areas. This area handles the packaging that bundles and protects the primary package. Secondary packaging such as labeling, cartooning, and inserting leaflets, package insert or PIL, in addition to track and trace serialization. Tertiary packaging such as bundling finished cartons into larger boxes, shippers, and palletizing for transport in addition to track and trace aggregation. Particular attention should be given to minimizing the risk of cross-contamination, mix-ups, or substitutions. The environment's still controlled, but with slightly lower stringency than the manufacturing and primary packaging areas. Focus on dust control and preventing mix-ups and the area's classification considered as CNC, controlled not classified. Production areas should be well lit, particularly where visual online controls are carried out. Quality control laboratories. Quality control laboratories should be separated from production areas, in particular those used for in-process controls, can be located in production areas. Laboratories for the control of biologicals, microbiologicals, and radioisotopes should also be separated from each other. Control laboratories should be designed to suit the operations to be carried out in them. Sufficient space should be given to avoid mix-ups and cross-contamination. There should be adequate suitable storage space for samples and records. Separate rooms may be necessary to protect sensitive instruments from vibration, electrical interference, humidity. Special requirements are needed in laboratories handling particular substances, such as biological or radioactive samples. Quality control labs in the manufacturing site can be replaced by outsourcing QC activities. Ancillary areas. Rest and refreshment rooms should be separate from other areas. Facilities for changing clothes and for washing and toilet purposes should be easily accessible and appropriate for the number of users. Toilets should not directly communicate with production or storage areas. Maintenance workshops should separate from production areas. Whenever parts and tools are stored in the production area, they should be kept in rooms or lockers reserved for that use. Animal houses should be well isolated from other areas, with separate entrance, animal access, and air handling facilities. Utilities. Without utilities, the entire pharmaceutical manufacturing operations grind to a halt. Now let's go deeper and look at the main 6 utilities that power these processes. 1. The HVAC system is arguably the most critical system. Clean rooms should be supplied with a filtered air supply through adequate heat, ventilation, air conditioning, HVAC, and ducting, which designed and constructed to minimize risks of contamination and cross-contamination. HVAC system has the following key roles. A. Controls temperature and humidity to ensure product stability and operator comfort. Provides filtration through pre-filters, bag filters, and ultimately HEPA filters that remove over 99.97% of particles as small as 0.3 microns. C. Maintains a positive pressure and/or an airflow relative to the background environment of a lower grade under all operational conditions and should flush the area effectively. Adjacent rooms of different grades should have an air pressure difference. D. Provides air changes, constantly flushing the room with clean, filtered air dozens of times per hour. 2. Water treatment unit. Water is the most used ingredient in pharma, but not just any water. Purified water, PW, used for the first rinse of equipment and in syrups and tablets. Made by processes like reverse osmosis and deionization to remove impurities and ions. Water for injection, WFI. This is the gold standard. It must meet strict pyrogen, fever-causing limits. It's typically produced by distillation or a specialized RO process and is circulated in a sanitized, continuously looping stainless steel system at high temperature, 80 degrees Celsius, to prevent microbial growth. 3. The chiller, where all that HVAC and some manufacturing processes generate a lot of heat. The chiller plant, often located outside, removes this heat by circulating chilled water throughout the facility to maintain that precise temperature control we talked about. 4. Boiler, the power of steam. Boilers play a crucial role in the pharmaceutical industry by providing steam for sterilization, heating, and maintaining strict hygiene standards in manufacturing processes. Steam is vital here. There are 2 types of steam used in the pharmaceutical factory. Plant steam, for humidification in HVAC and general heating. Pure steam, clean steam, generated from purified water. It's used to sterilize equipment and autoclaves that enter sterile areas. It leaves no chemical residues. 5. Compressed air is everywhere. It powers instruments, controls pneumatic valves, and even operates equipment in clean rooms. It's critical that this air is oil-free and dried to prevent contaminating the product or damaging sensitive instruments. 6. Drains. Drains should be of adequate size and have trapped gullies. Open channels should be avoided, but if necessary, they should be shallow to facilitate cleaning and disinfection. Sinks and drains should be prohibited in grade AB areas used for aseptic manufacture. In other areas, air breaks should be fitted between the machine or sink and the drains. Floor drains in lower-grade clean rooms should be fitted with traps or water seals to prevent backflow. If you found this tour insightful, please like this video, subscribe to Pharmatech Insights, and hit the bell icon so you don't miss our next deep dive into the world of pharmaceutical manufacturing. Thanks for watching.